of the NF-kappa B signaling in the nuclei, in which jellyfish collagen stimulate the expressions of NF-kappa B P65 compared to that of control. So this finding show us that jellyfish collagen will also stimulate the translocation of the NF-kappa B from cytosol to nuclei. So in other data, we may see that the in MAPK signaling, jellyfish collagen also stimulated the phosphorylations of JNK and also phosphorylations of PP38. So taken together, we may conclude that jellyfish collagen could stimulate the productions of cytokines, in this case is IL-12, by also inducing the expressions of the phosphorylations of I-kappa B alpha protein, just inducing the translocations of NF-kappa B from cytosol to nuclei. And in other hand, it also induced the activations of JNK and P38. So we may conclude that jellyfish collagen is a very potential substance to be used to stimulate the immune system and be used for functional food. That's all from me. These are the references, and thank you so much for your attention.
Okay, thank you very much, Pak Naro, for a very interesting presentation. We'll go to the next presenter, Bu Nina Herlina. Please play the video of the presentation of Bu Nina.
Assalamualaikum warahmatullahi wabarakatuh. My name is Nina Helena from Research Center for Applied Geology. Today, I will present a paper entitled "A Review of Infectious Pathogens, Causes of Reproductive Failures in Bull Sperm." As an introduction, beef demand in Indonesia is increasing every year, and the factors that boosting the demand of beef product in developing countries, including the growth of population, urbanization, economic refinement, consumer preferences. It has been a government concern to enhance cattle population since 2001 through the Meat Self-Sufficiency Program, followed by a special import required pregnant cows. Unfortunately, up to now, Indonesia has not been able to meet the necessity. There has always been a gap. Artificial insemination is the technique to improve the beef cattle population, conception rate, and offspring performance. And the semen quality is the key success. But unfortunately, there are some pathogens that can contaminate sperm that will directly affecting sperm quality or indirectly affecting sperm productions. And the purpose of this paper is to describe the different types of infectious pathogens transmitted sexually or transmitted via bovine semen, which affecting the reproductive performance and infertility of bulls. There are 3 agents, semen-borne pathogens, that we can classify: viral, bacterial, and protozoal. And in Indonesia, there are 12 of it that should be free from semen contaminations. As viral pathogens, there are 3 agents: bovine viral diarrhea, infectious bovine rhinotracheitis, and enzootic bovine leukosis. Bovine viral diarrhea, classified as an important disease regarding animal health and economic impact. The virus replicates in the seminal vesicle, prostate gland, and epididymis of the bull. And the detection of virus in testicular tissue might result prolonged testicular infection with detections time for 7 months following an acute infection, and artificial insemination with contaminated semen from acutely or persistently infected bull may induce an infection in female cattle. And infectious bovine rhinotracheitis is the virus can cause clinical disease of the male reproductive tracts, which is called infectious melanoprostitis, and it can replicate in the mucosa of prepuce, penis, and the distal urethra, and can be present in the seminal plasma of infected bulls. And angiotic bovine leukosis is the causative agents of angiotic bovine leukosis. Infected animals can transmit the virus horizontally via secretions contact, iatrogenically by a contaminated instrument, and the virus is rarely found in salmon because of through possibly present in association with virus-infected lymphocyte in the genital tract. And the bacterial pathogens is there are 1, 2, 3, 4, 5, 6, 6 of it. Brucellosis, septicemia epizootica, bovine tuberculosis, leptospirosis, paratuberculosis, and campylobacteriosis. Brucellosis is the infection can lead to orchitis, epididymitis, seminal fasciculitis, ampulitis, and decreased libido and infertility. The organism can also be present in collected semen. Septicemia epizootica can cause hemorrhagic septicemia in cattle. Bovine tuberculosis is a zoonotic risk and considerable economic losses. Paratuberculosis is occur in the intestinal tracts of cattle and other domestic and wild ruminants. And leptospirosis, the disease, is one of the major influences on the leptospirosis. And the campylobacteriosis caused by Campylobacter fetus subspecies venerealis. The organism resides on the penile and preputial mucosa of infected bulls, causing no clinical signs in the male. And the BGC is characterized by infertility and abortion in cattle. Protozoal pathogen is only trichomoniasis. Bovine trichomoniasis is a parasitic disease caused by Trichomonas petus. There are no specific clinical signs in infected bulls, except a preputial discharge associated with small nodules on the preputial and penile membranes in the early stage of infections. The infected animals may be carriers for their whole life. And the parasite resists in the diluted semen and to freezing. And diagnosis and control, the most popular is detecting and identifying bacterial clinical specimen by genotype. Control of pathogen of sexually transmitted disease can be done continually during blood and semen collections. Good health and physical status of bulls should be maintained properly. Biosecurity and protocols become necessary to reduce the probability of introduction diseases. Conclusion, the semen quality examination is a crucial step in the selection of bulls for the purpose of artificial insemination. This technique increasing the productivity and profitability through better conception rate and better generation performance. And this review helped to describe sexually transmitted pathogens, how to diagnose and control the disease so that the productivity and profitability of a particular commercial herd can be enhanced. Thank you very much.
Okay, thank you, Bu Nina, for your nice presentation. And then we will go to the third presenter, Pak Joko Triwibowo. Please play the video.
Hello everyone, thank you for the opportunity. I would like to present my talk with the title, The Potentials of Cultivable Sea Cucumber-Associated Bacteria. Today's talk will be divided into 5 parts. started with introduction. Sea cucumbers are marine invertebrates in Echinodermata. Sea cucumbers have many regional names such as Holoturian, Trepang, Tripang, Namako, Klingkau, Besedeme, Haitian or Gamat. Based on traditional Chinese references, Bechao Gang Xi and Zhong Yi Xiliao, sea cucumbers can be used as tonics and effective remedies to overcome health problems such as erectile dysfunction, constipation, rheumatics, hypertension, asthma, and even wounds. Further studies on their pharmacological activity show that sea cucumber have anticoagulant, antithrombotic, antitumor, antifungal, anti-leishmanial, until influencing the metabolism of lipid and carbohydrate properties. Sea cucumbers have potential components for functional foods and nutraceutical. Moreover, they also have potential components for cosmetics. Besides useful for the human's health, sea cucumber also important for the environmental health. Because sea cucumber playing a major role in the bioturbation of sediment, reducing organic load concentration in the surface sediment, enhancing the productivity of benthic biota through excretion of inorganic phosphorus and nitrogen, local buffering of ocean acidification, enhancing ecosystem biodiversity, and transferring animal tissue and nutrients which derive from detritus and microalgae to higher trophic level. Sea cucumbers redistribute huge amounts of sediments via ingestion and excretion, ranging from 9 to 82 kilograms per individual per year. The ingested sediments are rich in organic matter as well as bacteria. But bacteria are not the primary food source for sea cucumbers. Most of the associated bacteria in sea cucumber are transient. However, some bacterial species may be associated more permanently, especially in some parts of the intestines. Therefore, bacteria associated with sea cucumbers are an interesting study topic. But compared to other marine invertebrates such as sponges and tunicates, the studies on the biotechnological potential of sea cucumbers associated bacteria are still sparse. With most of the microorganisms not cultivable, this talk only focuses on cultivable bacteria from sea cucumber, published from 2007 to 2022. As we can see in the graph, in total there are 11 studies of sea cucumber-associated bacteria with bioactivity such as probiotic, anti-cancer, and anti-infective, which will be discussed further in the next slides. All of the bacteria with probiotic activity were isolated from sea cucumber Aposticopus japonicus. Aposticopus japonicus is an important commodity that can be found in East Asia countries. The bacterium Bacillus subtilis T13 improved the immune response of the juvenile sea cucumber Aposticopus japonicus when challenged with the pathogenic bacteria Vibrio splendidus. The consortium of the bacteria Pseudoalteromonas eliacovi HS1, Shewanella japonica SS7, and Vibrio tasmaniensis SS10, improving the growth, immune responses, and survival rate of sea cucumber Aposticopus japonicus. Another bacterium, Bacillus subtilis, showed probiotic activity by enhancing growth, immunity, and intestinal microflora of Aposticopus japonicus. There are several compounds that have been isolated from Pseudonocardia sp.SS7, which is an actinobacterium isolated from cloacal aperture of sea cucumber Holoturia maybii. These compounds were curvularin macrolides, which showed anti-cancer activity against several cancer lines, 4 glioma cell lines, and 2 colorectal cancer lines, with various IC50 values ranging from 0.59 to 81 to 0.01 micromolar. The mentioned compounds, especially compounds 1 and 2, also showed antibacterial activity against Escherichia coli with MIC value of 20 microgram per milliliter. Another study, which isolated Bacillus and Klebsiella from coulommic fluid of Holoturia atra, showed that those bacteria showed antibacterial activity against Salmonella typhimurium, Klebsiella pneumonia, E. coli, and Proteus vulgaris. And then another study, which is from our previous study, we isolated cyclodepsipeptides compounds from Streptomyces sp. SV21. These actinomyces were obtained from Stichopus vastus. These compounds showed antifungal activity, and in phaenomycin and also streptodepsipeptide SV21, they also showed antibacterial activity. The interesting thing is their antifungal activity almost similar or even stronger than the positive control. The conclusion and the future perspective from sea cucumber-associated bacteria is that the exploration of the biotechnological potential of cultivable bacteria from sea cucumber as probiotics, anti-cancer, and anti-infective resulted in the potential and some novel compounds. However, the studies were only from few species of sea cucumber such as Holoturia atra, Apostropus japonicus, Holoturia mebii, and Stichopus vastus. There are more than 1,500 species of sea cucumber that await to be further explored, especially on their cultivable associated bacteria. That's all my talks, and thank you for your attention.
Okay, thank you very much for your presentation, Pak Joko. So we will go to the, I think, last presenter maybe for this session is Pak Afif Haryawan. So please play the video.
Hello everyone, my name is Afif Pratama as a presenter of our research with title Bioinformatics Study of Active Compound of Morin Elboberry, Morus alba as breast anti-cancer. Breast cancer is one of the biggest causes of death in women. Statistically, in 2020, showed that breast cancer in women is the most diagnosed cancer with 11.7% cases, followed by lung cancer and colorectal cancer. Current cancer treatment show many side effects, including nausea and vomiting, bone marrow failure, and lymphedema. Herbal medicine that can affect the process of induction of apoptosis and inhibit the process of angiogenesis and cell proliferation make herbal medicine one of the potential in treatment cancer. Mulberry, Morus alba, is one of the herbal plants that is often used traditionally to treat cold, malaria, hypertension, and etc. Morus alba extract was reported in vitro have anti-cancer activity on several cancer cells, including breast cancer. Morin is one of the active compounds of Morus alba extract. Until now, information of the molecular target of morin, especially in breast cancer, is still limited. Morin has recently been reported to discuss the secretion of MMP9 and inhibit the active signaling pathway in breast cancer cell. In addition, there have now been publication targeting specific protein in breast cancer from morin. Molecular targeting profile of morin in form of energy activity and type of bond also need to be known in order to predict the resulting of biological activity of morin. Initial screening in exploring the potential of of other molecular targets of morin as an anti-cancer treatment can be done using bioinformatic method. Bioinformatic method has become one of the methods to discovery of new drugs because it can carry out an existing database-based approach. Method and result: breast cancer regulatory genes were collected with NCBI, and we get 5,237 breast cancer regulatory genes. Direct target protein or DTP are collected with stage, and we get 10 DTP from morin. Indirect target protein or ITP are collected with string, and we get 134 ITP from morin. DTP and ITP of morin were compared with breast cancer regulatory genes using 22.1. The overlap genes obtained are considered as protein target therapeutic genes or PTTGS. We get 96 morin PTTGS. This is the PTTGS of morin. And then gene ontology, pattern, and drug association analysis were performed using web gestalt by entering the PTTGS from morin. This is the result of biological process, cellular component, and molecular function of morin. You can see in the table. And then pathway enrichment. Pathway enrichment was used to determine the signaling pathway associated with the anti-cancer effect of breast. Morin can regulate 107 signaling pathway. The 10 signaling pathway associated with cancer can be seen in the table. Drug association analysis was performed to predict drugs that have a similar mechanism of action with morin in terms of gene target similarity. The result show 54 drugs had the same target with morin. The protein-protein interaction of Morus alba can be visualized using string database. And then the protein-protein interaction, we reanalyze using cytoscape, using the cytohuba plugin and calculated with the degree parameter to get top 10 hub gene. And this is the top 10 hub gene with the highest degree score are Caspase 9, TP53, AKT1, MIPK3, Jun, TNF, MYC, MIPK1, beta, and Caspase 9. However, there are some proteins that cannot be used as gene target, which increase the apoptotic process if targeted or inhibited. If targeted and inhibited, will cause disruption of apoptotic process. MYC and Jun cannot be used as target for drug therapy because according to the professor research, Jun and MYC cannot provide a definite antagonist effect. Structure of morin and protein, AKT1, MIPK3, and MIPK1, taken from protein data bank, prepared using ChemDraw and Autodoc tools by removing the water molecules and separating the native ligand to prepare the validation process. The validation process is done using PMOL software to see the RMSD value. But RMSD value is less than 2 Armstrong. The result of validation we can see in the table. After validation, we docking morin compound to AKT1, MIPK3, and MIPK1 receptor and see the visualization with the ISIBio. Morin was successful docked to AKT1, MIPK1, and MIPK3. In AKT1, morin exhibit less energy than the native ligand. The bond energy of morin, native ligand, and neratinib are min 7.6, min 8.2, and min 7.4 kcal per mole. This occurs because morin does not bind to Leu 156, ISN 279, and glycine 157, which are the active site of on AKT. In MIPK1, morin exhibit less energy than the native ligand. The bond energy of morin, native ligand, and neratinib were min 8.4, min 8.7, and min 7.5 kcal per mole. It's of course because Morin does not bind to level 105, which is the active site of MMPK1. In MMPK3, Morin showed a better bond energy than the native ligand and the actinium, which with each bond energy value min 9, min 8.3, and min 2.8 kcal per mole. Morin binds to serine 170, ISN 171, and cysteine 183, serine 170, ISN 171, and cysteine 183 were active site on MMPK3, which is not occur in positive control. Morin also banned valine 56, leucine 173, and alanine 169, which are important site in the binding of compound and ligand. This is just that Morin could be a potential compound inhibiting cancer by targeting MMPK3. The docking visualization of Morin with AKT1, MMPK3, and MMPK1 can be seen in this figure. Morin can inhibit cancer growth by binding to MMPK3 protein, which can active several transcription factors, namely CRAB, CMYC, and NFK-kappa B, which promote cell proliferation and enhance anti-apoptotic effect. Morin can target AKT1, MMPK. Conclusion, Morin can target AKT1, MMPK1, and MMPK3 protein. The docking simulation results the Morin being tethered to MMPK3 with a binding energy of min 9 kcal per mole are bound to the amino acid of serine 170, ASN 171, and cysteine 183, which are the active site of MMPK3. Thank you.
Okay, thank you very much, Pak Afif, for your presentation. And I just want to confirm to the committee whether the last presenter, Bapak Wail Adil, is he attend this session or not? Because I don't have the presentation material. If you don't have, then maybe I will continue to the discussion session.
Okay, so I think we can continue to the discussion session.
Okay, thank you. Okay, so Pak Afif will be the last presenter on this session. So we will continue to the discussion session. We have around 20 minutes. So is there any question from the audience? You can raise hand or you can type your question in the chat box. So this is one question from Pak Joko, right? Okay. I would like to ask some question to Mr. Abinaro Putra. How did you purify the jellyfish collagen? What is the difference between jellyfish collagen with other collagens? So, Pak Naro, please respond to this question.
Yeah, thank you. Thank you so much, Dr. Dini. Thank you so much, Dr. Joko, for your question. Actually, the one who prepared the sample is in another team. Yeah, I mean, our team were divided with several sections, but at a glance, how to purify the jellyfish collagen, actually most 80% or more the protein that could be collected during isolation process of jellyfish collagen, 80% is collagen. Okay, the protein that 80% of the protein isolated during the isolation process is collagen. So it means it is high enough to conclude that our sample is mostly collagen derived jellyfish derived collagen. So how did this be determined by our team is by using ion chromatography. and also by seeing the band pattern after separations using a SDS page. So did I answer your first question, Dr. Joko?
Dr. Joko, maybe you can respond.
Yeah, that's the first question.
I think so.
It's already answered the question?
Yes, I think so.
Oh, okay, thank you.
Okay.
So maybe I would like to ask
The second question, Bu. Sorry, the second question.
Second
from
question.
Dr. Joko. Yeah.
Okay.
What makes jellyfish collagen differ from other collagen? So the first one from the source, yeah, it is a marine collagen. Okay, marine collagen tend to be having a lower how to say denaturation temperature compared to terrestrial collagen. That's the first. And then the second, jellyfish collagen is, if I'm not wrong, sorry, I really forgot about this. It is categorized as type 2 collagen. This jellyfish collagen, and how to say the collagen has a specific amino acid conformations, glyXY, yeah, glycine X, blah blah blah, and Y, blah blah blah, and jellyfish collagen high in, if I'm not wrong, sorry, I forgot. I didn't expect this question appears. Yeah, it is also high in hydroxyproline. I think that's what I know about to answer your questions. Thank you. Hope it explains. Thank you.
Thank you for explanation, Pak Dr. Naro.
Thank you so much.
Okay, Pak Naro, I would like to ask, the jellyfish is indigenous from Indonesia species or no?
Thank you, Dr. Dini. The sample that we use, the sample that we present in this seminar was Nemopilema nomurai derived jellyfish collagen. So Nemopilema nomurai has been known by people in East Asia very well, yeah, including people of Korea, China, Japan. So this jellyfish is categorized as a giant jellyfish, and it is edible. It is edible. So most of the jellyfish that can be found in Indonesia is small jellyfish, stingy, and a little bit poisonous. I don't know, because maybe that's the reason not so many Indonesian people consume jellyfish. So the sample that we presented in this occasion is jellyfish that is mostly exists and can be found easily in Japan Sea. So maybe I could say it is native to East Asian countries.
So do you think our jellyfish can be potential also for producing the collagen or something?
I think yes, because the demand for halal and safe collagen is increasing year by year. So I mean, I don't know, Dr. Nina Herlina can be agree or not with me, but there is a concern using a terrestrial or land animal collagen because of the disease and so on and so on. So that's just a concern of people. I don't know it matters or not. Yeah. So to answer Dr. Dini's question, I think it's very potential to the flow Indonesian jellyfish derived collagen or any products for cosmetics or nutraceuticals. Yeah, it should be researched. Yeah.
Okay. Is there any other question? Thank you, Pak Naro.
Thank you so much, Dr. Dini.
Yeah, okay. Is there any other question from audience for Panaro or other presenters today? You can type in the chat box or just raise hand if you have question. If not, then I would like to ask Bunina. So you review about pathogen in the cattle. So how to identify the pathogens since it's so many, diverse pathogens that cause sperm contamination. Is there any detection kit or something to identify whether it's bacteria or protozoa or any species? Bu Nina, please.
Thank you so much, Dr. Dini, for your questions. Actually, our review is to identify and to describe the pathogens that should be free from sperm for artificial insemination. So there are 12 that classified as virus, bacteria, and protozoa. And the detections, since it should be to from BIB, Balai Inseminasi Buatan, so they use PCR to detect what is the contaminations and is there any contaminations from the semen. So I just want to write what is the pathogens that should be free from semen as artificial inseminations for cattle.
Oh, okay, so usually people identify by PCR, so whether it's a virus or a bacteria. So just using what universal primer for each target, like to identify bacteria, maybe 16S bacteria primer, like that.
Yes, PCR and ELISA.
PCR and ELISA.
Yes.
So for ELISA, how to do the identification?
There's a kit.
Okay.
Kit. So
ELISA kit for identifying the pathogen.
Yes.
So it's protein based or something? The kit? Okay. Okay, thank you, Bu Nina. So is there any other question from the audience? Okay, if not, then I would like to ask to Pak Joko. Is there any study reported about the diversity of bacteria in sea cucumber? Because your study is only about cultivated bacteria, if I'm not wrong. So is there any study reported about the diversity? And maybe my second question is, how can you access, how many actually bacteria can be accessed by cultivated approach? Okay, Pak Joko.
Thank you for your question, Dr. Dini.
Yes.
So can you hear my voice clearly?
Yes, yes.
Okay, so for your first question, is there any study regarding the diversity of bacteria from sea cucumber? The answer is yes. There are so many studies that has been done to identify the diversity of bacteria from sea cucumbers. There are some studies that I remember. First, they identify the sea cucumbers bacterium, the sea cucumbers bacteria, the diversity of sea cucumber bacteria, and they compare it to the bacteria from their surrounding environment, from their sediment. And the finding is, I found that it is interesting that the sea cucumber associated bacteria are different with their bacteria from the surrounding. So it means that the bacteria in sea cucumber, they are specifically stays there, even though, as we knew from the presentation, that sea cucumber ingests sediments up to 82 kilograms per year. So even though they ingest so many sediments from their surrounding, but their composition of bacteria is different with their surrounding. And then there is another interesting study regarding the diversity is from the ponds. So the sea cucumbers were cultivated in the ponds, and it is interesting to see that the diversity of bacteria from the sea cucumber, which are healthy in the ponds, are different with the sea cucumber that are sick. So sometimes the sea cucumbers are very populated in this pond. So because of the populated and then because of the rise of sea temperature, the pathogens bacteria are dominated of the diversity from this sea associated bacteria. And for your second question, sorry, can you repeat your second question, Dr. Dini?
So, I mean, how much we can access the diversity using only cultivated, because I think standard cultivation technique cannot access all the species in the environmental samples like sea cucumber. Yeah,
that's true. I agree. Yeah, so from my experience, I didn't compare the diversity of sea cucumber associated bacteria in both cultivable and non-cultivable with the cultivable one. So I'm not sure the exact number of the comparison between cultivable and non-cultivable ones. But as we know that we can only cultivate less than 1% of the total bacterial species that we knew from the diversity of bacteria in the world. Yeah, I hope it's answer your question.
It means that maybe so many potential bacteria that actually we don't know yet in the sea cucumber. So because I know other approach, so people usually extract first the compound and then try to find actually which bacteria who have that compound from the marine organism, like that. Yeah. So sometimes like that, not like your approach. Maybe you just isolated the bacteria and then try to find the potential of the isolated bacteria, like that. Maybe Pak Joko. Okay, this, I think in the chat there's a question for you, I think, maybe. And I don't know from who. Sorry, because it doesn't mention the name. Was your main reason to explore the pharmacological activity of sea cucumber associated bacteria instead of exploring the sea cucumber itself?
It was the question from Dr. Naro. So thank you, Dr. Naro, for the question.
Okay,
so
please.
I answer the question why I interested more in the sea cucumber associated bacteria, because first, there are a lot of studies regarding the sea cucumber itself. But why I interested in the sea cucumber associated bacteria? Because we can cultivate in unlimited amount of the bacterium, and once we get the bacterium, we can reproduce, or we can study it further without having to go to the field to take another specimen. If we depend on the sea cucumber, for example, and I want to get more the sea cucumber because I have interesting compounds, then I have to go to the field. I have to isolate the compounds from sea cucumber. And as we knew that we need a lot of biomass to isolate the small amount of active compounds. Take, for example, the ectenocidin from Ectenacide turbinata. We need about 1 ton or 1,000 kilograms just to get the amount of I think it's 100 milligrams of actinicidin. So it's a huge amount of biomass from the animals just to get the pure active compounds in small amounts. Yeah, I think it's more to the sustainability why I am interested to sea cucumber society bacteria. That's my ultimate motivation.
Okay. Panaro, it's already answered, right?
Yeah,
The
yeah,
question.
thank you.
Okay. So is there any other question from the audience for all presenters? If not, then I would like to ask the question to Pak Afif. So you did the molecular docking for the marine compound. I would like to ask, is there any follow-up after this bioinformatics study? Whether are you going to engineer the marine or something? Pak Afif, could you answer my question?
Okay, thank you for the question, Dr. Dini. I think I will follow up the research with molecular dynamic study. And then because this molecular docking study just show the protein and the compound in static movement, and then we must know how the interaction about the compound and the protein in dynamic movement. That's
okay. So it's still prediction, right? So it's
Yeah.
not proven yet in the lab like that for your bioinformatics study.
Yes, this is the maybe this is one of the prediction that before we want to study the new drug discovery.
But it's actually just one compound, right? It's only marine.
Yes, it's only marine.
Do you? So your method can be used for other compound or something with similar protocol. Your protocol can be used for other compound.
Yes, it can be used to search for another compound, like in Morus alba, like Morusian. We can use this method to know the molecular docking.
Is this new method, or you established this method newly, or it's already available, the method for predicting the compound, potential compound?
This is not the new method, but this is I use this method from Mr. Adam from Gajah Mada University.
Okay, Pak Afif. We still have time. Is there any question from other audience?
Yes, Miss Dr. Dini, if possible.
Oh, okay, please, Panaro.
Yeah, because you asked question to Pak Afif, I would like also to add question to Pak Afif. So I saw from your presentation that the compound marine, right, could interact with mitogen activated protein kinase 3, MAPK3, right? Correct me if I'm wrong, yeah. So this marine will inhibit the activations of MAPK3 because MAPK3 is one of protein that play important role to regulate cell cycle or to regulate growth of the cells. Correct me if I'm wrong. Yeah. Okay. However, this is my comment. MAPK3, or is also known as ERK1 or ERK2, is also expressed in many cells in our body. So if we inhibit this MAPK3, I am afraid, according to your suggestion, we also inhibit the growth of other cells in our body. So what is your comment about this? Thank you so much.
Okay, let me answer. So in my study, maybe it didn't describe what the specific cell that can be inhibited. So we in my study, we must to continue this study to know what is the molecular dynamic, and then what is the maybe if we want to discover this compound to be new drug, we can continue to in vitro study. Maybe that's my answer.
So maybe this is one of the way just to predict before we do the wet lab experiment, right?
Yeah, it's one of the.
Okay, so maybe you can make it not too broad like that. Then we don't know anything about the potential of our compound. Maybe it's like that, right? So it's just to make your research a bit narrow, not so broad maybe like that, yeah. By studying the molecular docking, using molecular docking, right? Pak Afif, maybe
Yes.
like that.
Yes.
Okay. So we actually has still many times because we should have 5 presenter, but only 4 presenter. So is there any other question to the presenters? If not, then I think I will close this session. So thank you to all presenters with very nice and interesting presentation, and also to all audience of this session. So we can give applause to all participants for this session. So Bapak Ibu, maybe you can have coffee break, and then you can join in the main room for the next parallel session. So thank you very much for your attention and contribution to this session. And Assalamualaikum warahmatullahi wabarakatuh. So I give back to the committee. Thank you.
Thank you so much, Ibu Dini, for moderating this session. So I
You're
think
welcome.
you, as Ibu Dini said, that you can go back to the main room for having a coffee break, because at the 3 p.m. we will still have a plenary session. So please join if you have your time. Thank you so much.
Recording in progress. My name is Baso Didik Hikmawan. I would like to say thank you so much to you all who have given me chance and time. So today I could present the result of our study about determining optimum harvest time of Corycistis sp for its maximum antioxidant capacity and pigment production. This is the background of our research. The natural antioxidants have become scientifically interesting for researchers due to its beneficial health effects. Well, natural antioxidants are used in pharmaceuticals, cosmetics, and also food industries. Therefore, the demand for natural antioxidant is rising. Most commercial antioxidant products are derived from plants. Unfortunately, destructive harvesting typically leads to the resource exhaustion and potentially species extinction for medicinal plants. Sources of antioxidants. Macroalgae are seen as one of a potential. Also, generation times depend on the concentrations of secondary metabolites such as carotenoid, phenolic compounds, and pigments. Harvesting macroalgae in exponential or high cell biomass actually reflects. So, in this study, we use Corycistis sp as one of potential macroalgae, and this study set out to identify the precise Corycistis sp growth phase at which the harvested macroalgal biomass concurrently has the highest antioxidant capacity and pigment concentration. Well, Corycistis sp was isolated from freshwater lake in Bengkulu Province, Indonesia, and cultivated with continuous aeration 6 media. Is mixed with Corycistis sp extract and measure it in spectrophotometer at 734 nanometer from this equation to determine its growth phase, which later used to observe the optimum harvest time from using hymocytometer and also using PD, a hot water extraction technique, which is dried biomass suspended dried biomass. This is the result of our study. As you can see in the screen, that we draw the growth curve of Corycistis. There were egg phase, sp stationary phase, then. So we use this. So this is the result for the pigment measurement levels in each growth phase. As you can see, this is the lag phase, log phase, stationary phase, and the death phase. And stationary phase as well in the 2.09. Next, in the antioxidant capacity measurement, we found that the log phase showed the highest inhibitions. It's that the optimum harvest time for macroalgae Corycistis sp in order to obtain the highest pigment and antioxidant concentration was when the macroalgae reached the final logarithmic or exponential growth phase, which is day 5 of the cultivation. And at the particular growth phase, the antioxidant extract of Corycistis sp was able to neutralize 91.51 to 99.75% of radical cation ABTS. So the pigment concentrations, especially chlorophyll, was also in highest concentration at the final logarithmic phase. However, the carotenoid content was highest when the macroalgae grew until the final stationary phase or in day 10 of the cultivation. Therefore, if it was carotenoid that was expected to be high in the harvested biomass, then macroalgae, this Corycistis sp should be harvested when it was in the final stationary phase or in day 10. Well, this is the reference that biomass. Then macroalgae, this Corycistis sp should be harvested when it was in the final stationary phase or in day 10. Well, this is the reference that we use. And I say thank you very much for your attention.
Welcome back to the main room, ladies and gentlemen. The next agenda is plenary sessions for this afternoon. Therefore, I may invite Dr. Ira Handayani as the moderator of the sessions. But before that, allow me to read short profile of Dr. Ira. Dr. Ira Handayani is currently a researcher in Research Center for Applied Microbiology, National Research and Innovation Agency, the Republic of Indonesia, who are interested in molecular genetics, microbiology, and bioinformatics. She obtained her bachelor degree in chemistry at University of 11 Maret, and she continued her master degree in biochemistry at Bandung Institute of Technology, or ITB, and also her PhD in chemical engineering at Tübingen University, Germany. Dr. Ira, time is yours to lead the sessions.
Okay, thank you, Ms. Aulia. Are you hear my voice? Okay. Hello, are you hear my voice?
Yes, we can hear you
Okay.
clearly.
Okay.
Dr. Ira.
I don't know whether Yvonne already in here or not.
Yes, good afternoon, Ian.
Oh,
I
good
hear you, and
afternoon,
I'm
Yvonne.
already here.
Okay, how are you?
Thanks, fine. Hope you too.
Okay. Okay, thank you. Okay, I will start the presentation. The plenary session for this seminar. First of all, I'm very happy to see you here and welcome all of you to this plenary session. Before the presentation starts, let me tell you how the presentation will go. First, our first speaker, Professor Yvonne Mas, will be invited to present the material, and then our second speaker, Professor Claudia Tiribelli, will also be asked to present his material. After both speakers present their material, there will be a question and answer session followed by a conclusion. Now let's start with our first speaker, Professor Dr. Yvonne Mas. Okay, this is the CV of Professor Dr. Mas. Professor Dr. Mas is a professor of applied microbiology at the Technical University of Brunswick and head of the Department of Bioresources for Bioeconomy and Health Research at the Leibniz Institute, the SMZ, a German collection of macroorganisms and cell culture. She has experience as an editor in the journal Molecule and the journal Printer in Microbiology, and experiment as a member of the topical advisory panel in the Journal of Antibiotics. She is interested in research in the field of microbiology, biotechnology, actinomycetes, antibiotic, and regulation. Now allow me to welcome Professor Dr. Yvonne Mas to make her presentation. Professor Mas, please explain your material. And time is yours, Yvonne.
So thanks, Ira, for this nice introduction, and good afternoon to everybody.
Okay.
Okay, now I see my slides. Okay. Okay. So again, good afternoon to everybody. Yeah, thanks for the nice introduction. Actually, I'm here at the Leibniz Institute, DSMC, so the German collection of microorganisms and cell cultures in Braunschweig, Germany. And it's a great honor to give a talk during your session at the International Symposium of Innovative Bioproduction in Indonesia. And today I would like to introduce you into the topic of how we can exploit the biodiversity for novel natural compound production by using regulatory switches. So I should, now it's okay. So before I go into that more in detail, I first want to mention why this kind of research is relevant at all. And I think all of you know that antibiotic research indeed is relevant because we observe growing crisis from antibiotic-resistant bacteria. So the World Health Organization regards the antibiotic resistance disease as one of the 3 biggest threats to global health. So of course, it's urgently needed to find novel antibiotics to combat these drug resistances. But the question is where to find them. I'm sorry, the presentation is a bit slow, but I hope we can do that.
I think it's okay.
Okay. So we can, of course, find antibiotics a lot in actinomycetes. So actinomycetes are known to produce 2/3 of all antibiotic in clinical use today. And this antibiotic production potential is known for quite a long time. However, what is not known for such a long time is that the genetic potential for the production of antibiotics is even larger in these organisms. And this appeared when the first genome sequence of such an antibiotic producer has been sequenced. So this was the genome sequence of the model organism, Streptomyces silicolor. And that silicolor strain is known to produce up to 2 to 4 secondary metabolites under standard lab conditions. However, the genome sequence analysis revealed that the genome harbors up to 20 different biosynthetic gene clusters. However, not all of those gene clusters are expressed, meaning not all of these substances are really produced, but a lot of them occur as so-called silent antibiotic gene clusters. And this is not only true for that organism, but nowadays, since we have a lot of, more than hundreds of genome sequences available from different actinomycetes, it turned out that this is true for a lot of these producing organisms. So obviously, actinomycetes still hold a huge potential for the production of novel secondary metabolites. And of course, the current trend is to activate these kind of silent gene clusters. And now I would like to introduce you into the topic, how we can activate those kind of silent gene clusters in actinomycetes. Okay, and we actually can do that by using regulators. And regulators indeed are the set screw to unlock the biosynthetic potential of these natural compound producers. However, the problem is just that there are so many different transcriptional regulators abundant in actinomycetes, especially in streptomycetes, and these can be of different classes, and they can interact in quite complex cascades with each other. And depending on genome sizes, those can actually be more than thousands of different transcription regulators. So the question is, which is the most suitable and most potent type of regulator that is suitable to be used as a general activator of antibiotic biosynthesis. And today I will focus on these subtype regulators, since we think that these are good candidates to be used as general activators of antibiotic biosynthesis. Okay, presentation is still slow, but okay. So first of all, I will introduce you where we found this type of regulators. And we indeed identified them in the antibiotic producer Streptomyces pristinus pluralis, which is kind of our working horse. So this strain produces the pristinamycin substance, the streptogramin antibiotic pristinamycin. And pristinamycin is actually 2 different types of antibiotics, the peptide antibiotic pristinamycin 1 and the polyketide antibiotic pristinamycin 2. And the Streptomyces pristinus pluralis strain produces those 2 substances in a specific ratio. They interact synergistically with each other, and a semi-synthetic derivative of that substance is on the market. It's called Synergid, and it's used as an antibiotic of last resort against highly resistant pathogens. And what we did back then was that we characterized the antibiotic biosynthetic gene cluster. And back then, this was the largest antibiotic gene cluster that has been described so far. And the genes that you can see here in red, they are needed to, they encode for the peptide antibiotic pristinamycin 1, and the blue ones encode for the polyketide antibiotic pristinamycin 2. So when we had a closer look into this antibiotic gene cluster, then we identified a set of 7 different transcriptional regulatory genes, and they belong to different types of regulatory families. And actually, this is quite a large number, because usually these kind of antibiotic gene clusters have one or two such kind of regulatory genes. And we studied the function of all of those regulators in detail. However, today I will only focus on this red labeled PAPR2 regulator here, which belongs to the subtype family of regulators. So when we studied the function of this regulator more in detail, and there I need the next slide. Okay. First of all, we inactivated the gene and studied the pristinamycin production, and there we observed that the inactivation of the PAPR2 gene leads to complete loss of pristinamycin production in comparison to the wild type, which still nicely produced pristinamycin 1 and pristinamycin 2. So therefore, we knew that PAPR2 is an essential activator of pristinamycin biosynthesis. Ira, can I ask you, are you the one who is moving the slides forward, or can I do that?
No, I don't know.
Okay,
no,
because it's.
I think it's a bit left in there.
Yeah, it's really several.
I will ask Sandy.
But I will. It's okay. I will just move on. So PAPR2 is an essential activator of pristinamycin biosynthesis. This was what I wanted to say. And we studied the function of that regulator also more in detail, and figured out where the regulator really binds within the antibiotic gene cluster. So therefore, we purified the PAPR2 regulator and did electrophoretic mobility shift assays, and thereby we could show that PAPR2 really binds to the promoter regions in front of the pristinamycin 1 and pristinamycin 2 structural genes. And we were also able to define a specific consensus sequence for such a binding. And by RT-PCR experiments, which are shown here at the right-hand side, we also could show that there's no transcription of the pristinamycin 1 and pristinamycin 2 structural genes in the PAPR2 mutant in comparison to the wild-type organism. So therefore, we knew that PAPR2 is a direct activator and an essential activator of pristinamycin biosynthesis in Streptomyces pristinus pruritus. And the next slide, which I hope comes in a few seconds, shows you where the regulator is situated within the regulatory signaling cascade. Because it's here located at the very bottom, it directly activates the pristinamycin biosynthesis, and itself it is regulated by a set of other transcriptional regulators where I will not go in detail. However, it's quite complex signaling cascade governed by microbial hormones called gamma-butyronolactons. And what I wanted to mention here is that understanding these kind of regulatory signaling cascades and the regulation mechanisms of antibiotic biosynthesis in general can also help to activate silent gene clusters. And this we observed when we overexpressed the PAPR2 regulator in a foreign strain. And back then, this was Streptomyces lividans. And this lividans strain should come also in a second. This lividans strain usually does not produce any secondary metabolite under standard lab conditions. So this you can see here on the left-hand side, the lividans strain with the anti-vector control. However, when we overexpress the PAPR2 regulator, then the lividans strain produces an intensively red pigmented substance. And this red pigment indeed is andecyl protegiasin. And andecyl protegiasin is a known product from Streptomyces lividans. However, it is usually not produced in the lab, so it's encoded by a kind of a silent gene cluster. And this andecyl protegiasin biosynthesis is known to be under control of a subtype regulator called RED-D. And this RED-D shows quite high sequence homology to our PAPR2 regulator, so up to 55% sequence homology. And therefore, we speculated that PAPR2 takes over the function of RED-D and leads to the activation of this silent andecyl protegiasin biosynthesis in Streptomyces lividans. And we also later on could show that on the molecular level. So I'm really sorry that it's hanging, but yeah, this is what is shown in here. So having that in mind, we had a closer look into this kind of subtype regulator families in general. So in literature, they are described as streptomyces antibiotic regulatory proteins. They are known to act as pathway-specific activators of antibiotic biosynthesis. And all of them contain a conserved N-terminal DNA binding domain. And with this, they bind to conserved consensus sequences at the promoter DNAs of the promoter regions in front of the target DNAs. However, so far, the regulatory mechanisms, how those regulators really work, is totally unclear. So this is something what we are going to study in the near future. So we will do crystallizations of the regulator and try to figure out how this activation mechanism really works out in the antibiotic producers. So having that in mind, we had a closer look where those subtype regulators occur, and we found that the genes are very abundant in many different actinomycetes. So you can find them in nearly all streptomycetes, but also in other important antibiotic producers belonging to the actinobacteria family. And when you have a closer look where they occur within the genome, then you can find them as part of biosynthetic gene clusters. And actually, as part of gene clusters that encode for different types of secondary metabolites like beta-lactam antibiotics, phosphonate antibiotics, polyketides, and so on. So therefore, we knew, okay, the subgenes are abundant in very different actinomycetes. They are part of gene clusters encoding for different types of secondary metabolites, and therefore we said that those ones are good candidates to be used as general activators of antibiotic biosynthesis. And then we tested this out in a random strain in just a running project of ours, and this also will be shown, I hope, in a few seconds, because back then this was a project which we performed with some Indonesian partners. So actually several ones that are sitting in the audience today. So with Puspita Listianti, Vin Kusaryoto, Iran Dayan, Shantaratna Komala, and so on. So back then we aimed to isolate new antibiotics from Indonesian actinomycetes. So yeah, there we are. Back then, the Indonesian colleagues isolated up to 422 actinomycetes from Indonesian habitats. A subset of strains was handed over to our labs back then at the University of Tübingen, and we prioritized the strains for the most phylogenetically unique ones. We grew them in several different cultivation media and covered all of that to bioactivity tests to prioritize the one with the best bioactivity. And back then, 9 strains were of most interest, and from these ones we isolated the genomic DNA of, did biosequencing, and then performed a combined genome mining molecular networking approach. And I think this was what Ira mentioned some hours ago in her talk. So this we have done for dereplication purposes and focus on the most unique gene clusters that encode for the potential unique substances. And back then, one of the interesting strains that turned out from this analysis was SHP-227, which showed quite some good activity. And we just randomly used that strain and performed our sub-guided activation strategy with that. So we cloned in our PAPR2 overexpression strain to SHP-227 and got extracts of the cultivation of the strains and tested it against Bacillus subtilis as a test organism. As you can see in here, the SHP-227 PAPR2 overexpression strain showed significantly improved bioactivity against Bacillus subtilis in comparison to the control strains with the anti-vector or the wild-type samples, which means that PAPR2 overexpression led to significantly improved bioactivity of that SHP-227 strain. Now, what you would do in the next step is that you go into the lab and do elaborative isolation of that compound and check what the substance really is. But we didn't do that. In the first place, we started a genome mining approach of that strain in order to find out what might be the substance that encodes for this, or what might be the cluster that encodes for the substance with this improved bioactivity. And as I said, we have the genome sequence in hand. We performed an anti-smash analysis for the prediction of potential antibiotic gene clusters, and thereby we identified 25 potential secondary metabolite gene clusters. And then the question was, which is the one that encodes for this bioactive substance? And then we ran an additional bioinformatic analysis by applying the program PET scan and searched for our PAPA2 consensus sequence. And thereby we found this subtype sequence in front of gene cluster 9. And gene cluster 9 encodes for potential nucleoside antibiotic, which has similarity to a gene cluster from a Streptomyces lineaceus strain. And that one is known to produce the substances amicetin and plicacetin, which are known compounds. They act as protein synthesis inhibitors with quite good bioactivities. And only when we knew that, then we went into the lab and checked by HPLC analysis what the product of this strain really is. This should be shown in the next slide, hopefully in a few seconds.
Sorry, Yvonne, I'm interrupted. Your time is 3 minutes again.
Okay, I will hurry up.
Sorry.
And I will go through the slides. Okay. So back then, we indeed could show that it's amicetin plicacetin that is produced by the PAPA2 overexpression strain, and in significantly higher amounts. In the next slide, it is shown that this also helped for the isolation of 4 novel pyrimidine nucleoside antibiotics, which was a collaborative work with the group of Harald Groß at the Pharmaceutical Institute of the University of Tübingen. I'm really sorry, but the slides are just too slow. So this was just published in the beginning of this year. And now we know that this sub-activation strategy really works out. We are now having a set of different overexpression plasmids. I will skip that, but I hope, yeah, I hope that will work. So what we are just doing at the moment is that we apply this to the genome sequenced strains of the DSMC. So we scale that activation strategy up, and we already screened hundreds of genome sequenced DSMC strains for the abundance of sub-genes and sub-consensus sequences. And these are the ones that we are currently using for performing our sub-guided activation strategy. And in one of the next slides, hopefully, I still have the time that also, I will skip that one, that also in our selected DSMC strains, where we clone in our PAPA2 regulator, we observe significantly increased bioactivities in comparison to the control strains. So when we test different test strains as for the bioactivity. And for some candidate strains, we already have some potential substances that are produced. For several strains, you can already see by the eye that something is going on, that when you clone in the PAPA2 construct, that there's significantly production, that there's production of a pigment in comparison to the wild type strain. And we are currently isolating these substances and trying to find out if there's a novel antibiotic that is produced by those strains. And with this, I'm at the end. I'm really sorry that the slides were kind of delayed, but I would like to thank all the people that have been involved in this project, mainly Ira Handayan, who worked a lot with these Indonesian strains, especially with the SHP 27 strain. Also, Janina Krause performed a lot of these SARC overexpression analysis, and currently the right-hand person are involved in the SARC-guided activation strategy of the DSMC strains. And with that, I would like to thank for your attention. I hope we can move on with joint collaborations, because I really enjoyed that interaction with you. Okay, so thanks, and I would be happy to take questions later on.
Okay, thank you, Yvonne. Thank you very much for giving such an informative and exciting presentation. Now, let me invite our second speaker, Professor Claudio Tiribili, MD, PhD.
Here I am.
Okay. Professor Tiribili, okay. Okay, wait a minute. Okay, okay. Okay, thank you. Professor Tiribili is the scientific director at the Fondazione Italiano Fegato. He has
Italian Foundation. That's easier.
Oh, okay. Sorry. He has experience as an editor in the Journal of Hepatology and the Annals of Hepatology Journal, and experience as an associate editor in the BBC Research Note Journal. He is interested in research in bilirubin, neonatal jaundice, metabolic liver diseases, hepatocellular carcinoma, stem cell in hepatocellular carcinoma, Kriegler Nazar syndrome, and translation medicine. Now allow me to welcome our second speaker, Professor Claudia Tiribili, MD, PhD, to make his presentation. Professor Tiribili, time is yours to give a lecture now. Thank you.
Well, can you hear me?
Yes, of course. We can hear you.
Thank you very much for this kind introduction. Let me try to share the screen with you. That's already done. And so I can start my presentation. Let me tell you, it's really a pleasure to be a speaker of the 9th International Symposium on Innovative Bioproduction in Indonesia on biotechnology and bioengineering. What I'm trying to address today, I'm driving the presentation. What I'm trying to do today is actually make a link to what we are eating and drinking and the liver damage. As you know, no, it doesn't work. So why don't we remove my presentation? I'll share my screen.
Okay. Stanley, can you help me?
Yeah, I mean, I need to go on. Let me try to share my screen. And then, here we go. Okay.
So it's moved now?
Yeah.
Okay.
Can you see it?
Yeah,
okay.
I'm seeing it now.
Okay, let's move back to the start. Sorry for that, but okay. So liver is, after the skin, is the largest organ in our body, and in an adult weighs about 1.5 kilo. Although it's not that thick, the normal liver has a volume similar to the soccer ball. And during this period, we are very fan of a soccer ball because it's the largest gland in our body, and 80% is composed by the liver, by the hepatocytes. But most important, the liver is the organ in our body which is the largest number of functions. Liver can do more than 600 functions, such homeostatic, metabolic, detoxificant, and immunological function. But in addition to that, and not only a marginal issue, is that the liver is the only organ in our body able to reproduce and regenerate. And this is a Prometeo myth is being depicted. The main causes of chronic liver disease are different things, such as diet, here, medication, viruses, and autoimmune liver disease, moving from normal liver to usually fatty liver, which is increased presence of the fat in the liver, which can go on with the steatohepatitis and then go to liver cirrhosis, which is advanced liver fibrosis and functional complication, ending up with the presence of hepatocellular carcinoma, which is the end stage liver disease. we need to stop this trip from here to here. And what I'm going to address today mainly is the role of eating and the diet in having liver disease. So, and this is why, because MAFL, the so-called metabolic fatty liver disease, associated metabolic liver disease, has a prevalence globally of more 25% of the population, which means in one out of the 4 European citizen has a fatty liver, so has a possibility to develop a serious liver disease. So if we go from the trip of from normal liver to hepatocellular carcinoma goes via different steps. First of all, we have the MAFL, the non-alcoholic liver, alcoholic fatty liver disease, which is, mainly represented by the increased amount of fat into the liver cell. But once the inflammation occurs, due most probably to oxidative stress, we have a sort of activation of the extracellular matrix of the liver, which is able to create scares and then move from NASH to cirrhosis, which is, labeled by the presence of scars in the liver, making the liver hard and less active. And these scars, together with the regeneration of the liver, end up in the hepatocellular carcinoma, which, as I told you, is the end stage of this trip. We have several risk factors for this. Of course, we have the overweight, the obesity. I'm going to touch this in more details later on. We have type 2 diabetes and insulin resistance. Altogether, overweight or obesity, type 2 diabetes, and insulin resistance make what we know now is called the metabolic syndrome. And the metabolic syndrome is related both in adults, but even more worrisome, is present also in children. And this is something we have to think about, because being in children, we need to prevent the fact that our child moves from here to here in the life span he or she is expected. And if we look here, that's very remarkable figure, that if you look the prevalence of obesity from 1975 to 2016, both in females and in males, you may see the dramatic increase in the prevalence of obesity during this year, meaning that the more so-called civilized we are, the more fat we are going to be. And this is something we have to cope with and we have to fight with. And actually, even more worrisome is the trends in the number of children and adolescents with obesity. And if you both in males and in females, this is true all over the world. But if you calculate here, which is the Southeast Asia, also in the Southeast Asia, particularly in the male children are associated with an increased level of fat in the body. And if you look around the world, the presence of the metabolic fatty liver is basically almost the same all over the world. In some regions it's higher, like in US, in South America. In some regions it's lower, as Africa, due to the fact there is a reduction in the calories available. Australia is basically like Europe, but in China we are observing now a booming wave of obesity, and this is something we have to cope with. So going back to what, in my opinion, is the most important aspect of the obesity is the so-called vicious circle of childhood obesity. We have a child which actually due to a wrong life habits, sedentary life, lack of physical activity, is getting mildly obese child. And the mildly obese is actually a child is less prone to exercise, and those goes from mild to moderately obese child. And if this is also keep going with a wrong diet, is going to move from moderately obese child to severely obese child, and actually to and then to obese adult. And this is where we are going to go from a healthy child to a potentially sick child with the presence of fatty liver. And if you look around, you see that genetics may play a role in obesity. And we know now that the contribution of heredity to the risk of obesity is 25%. And this variability is related to the genetic background of the different studies performed. So we need to understand that habits, in addition to genetics, is the main predisposing factors, particularly habits of parents living with the child. And we just published an editorial in an international peer-reviewed journal, was APT, making clear that it's the parents giving us the wrong information how to behave, less exercise and much more sedentary life. What are the targets of fat accumulation in the body? Of course, obesity is a multi-organ and multifactorial disorder. We can have damage in the liver, in the brain, in the heart, in the vessels, in the nervous system, and in the pancreas, and in the muscles. Altogether, meaning that we have a lot of complications of obesity in the childhood. We have damages in the behavior, psychosocial, neurological, like pseudotumor. We have pulmonary, sleep apnea, asthma, and intolerance of the exercise. Of course, cardiovascular, due to the dyslipidemia, hypertension, coagulopathy, gastrointestinal, as I told you about steatohepatitis, and also renal, endocrine, and muscular. So this, we have to conclude that an obese children, like an obese adult, are sick, and we need to cope with. So which are the main causes? Is the imbalance between the calorie taken and the calorie expended. I am used to say that the weight is like our bank account. If you eat, if you gain a lot of money, you don't spend, you get rich. But if you eat a lot of calories, but you spend a lot, you get poor. So we need to understand that globally, there's a striking decrease in physical activity. 1 in 5 boys and 1 in 10 girls is not having a real and appropriate exercise. And even more worrisome is that the amount of fruit eaten by our children is drastically reduced as compared to the ideal amount. Only 30% of boys or roughly 40% of girls eat a fruit a day, in spite of the fact that fruit is readily available all over the world. And the major suspect is sugar. Sugar, the white sugar, it's really being associated with the presence not only of obesity, but also liver disease. And the question is how much is too much? And we need to understand this because we can give advertising. And here is what we have. And what we have here in the slide is coming from the World Health Organization, telling us the content of teaspoons for each beverage. And mind that the WHO recommends consuming no more than 12 teaspoons per day. If you drink a Coca-Cola, you drink it's like you eat 8.5 teaspoons of sugars. If you eat a yogurt drink, it's like 7. But if you need an energy drink, you have 10. And of course, telling you that what we are using to give our children, the so-called sodas, are then maybe very dangerous. And here's what we have in as comparison. Of course, the blank is the water. There's no sugar. But if you look at Sprite and Coke, which is widely used, we have that 33.7 and 35 grams of sugar per each can. And even worse are Fanta and Schweppes, when we go from 33 to 43 and 44 gram of sugar. So telling us that, we are not surrounded by a friendly beverage consumption. What is the total fat? The total fat should not exceed 30% of the total energy intake. And we have different kinds of fat. We have trans fats, the dangerous one, due to the so-called junk food. We have the saturated fat, which is related to coconut, palm oil, and alimentary, polyunsaturated skin, meat, and fruit. And we have then the unsaturated fat, which are the real good fat, that should be much higher than 90% of the total fat in intake we have every day. And this is important to prevent additional damage. How the excess of fat?
Sorry, Professor Tribuli, I may interrupt you because your time is only 4 minutes again.
Well, my time is still 5 minutes.
Oh, 5 minutes. Okay, 5 minutes again.
I mean, according to my watch, it's still 5 minutes.
Okay.
So I'll be on time. Don't worry.
Yeah.
So how the
Thank
excess
you.
of fat and sugar impact the liver? This is a cartoon. It's a very complicated cartoon. I've had no time, unfortunately, to go through. But the key issue is that insulin resistance and increased intake for the diet bring together the possibility to accumulate fat in our liver. And this is what we have to deal with. So do we have any models to study the juvenile fatty liver disease? And the answer is yes. This is work we have done in our lab, driven by Sylvia and Natalia and Pablo. And what they did, they reproduced the fatty liver, a juvenile fatty liver, in a mouse, giving the mouse a lot of fat and a lot of calories. And what we have, what they have been able to reproduce is a fully scaled presence of fibrosis, meaning telling that this may be a good model to deal with in order to understand if there is any good treatment to work with this. In addition to that, we know that fatty liver is really linked to a leaky gut, because the gut is living alone with a lot of toxins moving from the gut to the liver. And this is what we have observed, that in large intestine, we have a leaky, the presence of a less tight junction as compared to controls. So in conclusion, we can say that excessive sugar and fat since young age induce obesity-related multifactorial events that lead to progressive multi-organ dysfunction, fibrosis, leaky gut, altered glucose homeostasis, means diabetes. And this is, and we have to realize that the progression in children is much faster than adults. So we need to take care of this aspect for the future generations. And we know now that in the future 20 years, all the liver transplantation will be related to fatty children at the moment. So we need to take action on that. So let me skip all this because I have no time here. Let's try to deal with the future perspective. We heard that insulin resistant, type 2 diabetes, and obesity is the accounts for metabolic syndrome, which is a booming of obesity. And to date, we are still missing something, but we need to do something as a diagnosis, screening, and efficient treatment. And the clock is still ticking. So thank you for attention. I think I did my job in time, and I used all my 20 minutes.
Okay, thank you very much, Professor Tribuli. Thank you for giving us such an informative and interesting presentation. And ladies and gentlemen, now are the time for the question and answer session. We have 20 minutes for Q&A session. If you have question for both speaker, please raise your hand and or you can write the question in the chat room. Okay, I think we have wait a minute. I need to see. I think Miss Atika, are you ready to give your question?
Thank you, Dr. Yeah, my question is to Dr. Yvonne. She's gone. No. Yeah.
Okay.
So,
Yvonne, are you still in here?
I'm still here. I'm here.
Okay.
Yeah. All right. Okay. So my question is a bit practical, but I never done the analysis that what you did. So my question is like how long actually to get from your analysis of pre-synthetic gene cluster until the analysis of the mutant, if I'm not mistaken. It's like you did like cloning for all this experiment, right? If I'm not mistaken. So ideally, how long until you get the results? Thank you.
Yeah, thanks for the question. That's actually hard to say because it's mainly dependent on the strains to manipulate. Actinomycetes are not so easy to genetically manipulate, and depending on which kind of organism you have enhanced, this really depends how fast you are. But concerning the whole process, depending if you have the genome sequence, this is the first step. Let's say if you have the genome sequence, then you could go for the anti-mismatch analysis, and this is quite fast because you just upload the sequence on the anti-mismatch tool, and then you directly get the output after a few minutes. But this is, let's say, pure bioinformatics. It can give you an estimation what kind of gene clusters may be encoded, but you then need to really have a closer look into that and study that more in detail and also manually. And then, I mean, nowadays we have these overexpression plasmids enhanced, they are cloned, then you just need to introduce them into the actinomycetes. And if everything works well, you are maybe successful after 1 or 2 weeks. If it's a strain that is harder to treat, then it can take several months, I would say. But this is what where I skipped a slide, because what I wanted to mention is that nowadays we have different genetic tools. So we have different plasmids, which can be used either for protoplast transformation, which is a process how you can get DNA into the strains, or you can use the plasmids for conjugation, where you use the opportunity that you have an E. coli strain that can deliver the plasmid to the actinomycetes.
Okay, Yvonne, thank you for the answer. And in the chat room, I have 3 questions for Professor Tribeli. First question is, it is true that some herb like curcuma is help in maintain our liver health? Thank you. This is from Mr. Elok. Miss Elok, I think. Are you ready to answer?
Yeah, the answer is maybe. I mean, there's no real confirmation that any drug is preventing liver disease. There are some drugs used to or suggested to be helpful, but I mean, the real scientific demonstration is not yet. The curcuma is one. The other one is silymarine, the thistle syrup. Others are the extract from the apple. But I mean, there are some information, but not real specific and solid scientific demonstration. So we need to take this with a very, with caution.
Okay, thank you for the answer. Okay, and the second question is, what are the most recent and reliable treatment to deal with liver damage that are already on cirrhosis stage? Is surgery and transplantation are the only option?
Well, it depends about the stage of the disease. Of course, if you have an end-stage liver disease, transplantation may be an option. Actually, it is an option. We have to realize that in Europe, every year we are performing roughly 2,500 liver transplantation all over Europe. But I mean, the ideal is not to go to the liver transplantation. The ideal issue is to stop the trip before moving ahead. This is what I try to demonstrate in my presentation as far as obesity is concerned. If we start changing our lifestyles by asking our adolescents and ourselves to exercise more, to eat less, and to be much more pragmatic in terms of daily behavior, this could be very helpful. Not only for the liver, but also for cardiovascular disease, for diabetes, for everything.
Okay, thank you for the answer. And next, are there any possibility to recover from any damage in the liver?
For what? For?
Are there any possibility to recover from any damage in the liver?
Absolutely. I mean, liver is a regenerating organ. If you have a acute hepatitis, I mean, and you heal from the hepatitis, I mean, like a hepatitis, the liver returns back totally to normal structure. So we don't have to have any scar. Of course, if you don't recover fully, for instance, from B hepatitis or C hepatitis or other diseases, then the situation is not acute, it's going to be chronic. So the damage is chronic. So the end result is that the liver is daily damaged by the heating stimulus. And this ends up with something which is chronic liver disease.
Okay, thank you for the answer. And now we still have 13 minutes left. If there are any question for both of speaker?
We are being too clear.
Okay.
Or too confused.
Okay.
I have no idea.
If there any, if there's no any question again, I will ask maybe to Professor. Okay, Professor Mas. I want to ask Professor Iwan, what medium was used for self-activation with genome sequence in DSMZ strand? Is there any effect of the medium on the successful use of the self strategy in activating the silent gene cluster?
Yeah, actually, also here we tested a different set of cultivation media just to make sure that we do not miss anything. So actually, we tested 3 to 5 different cultivation media. Some of them you may know, I will not go in detail with these ones, but it turns out that if you have activation, that 1 or 2 cultivation media are enough. So it looks like that really the abundance of the subtype activator is more relevant than the media you use.
Oh, I see. Okay,
thank you.
Yeah,
but Ira, I think I also see a question in the discussion panel or in the chat room
okay. I will check
from Hatati. I may also read it by myself because the question was what kind of data that one inputs to get biosynthetic gene cluster using Plantis mesh. So I'm not sure if this is if I understand the question correctly, because there are different tools available. So AntisMesh is the program that can predict antibiotic biosynthetic gene clusters encoded in bacteria, and PlantisMesh is a tool where you can identify gene clusters from plants. So if this question really targets PlantisMesh, and the question is which kind of biosynthetic gene cluster one can find there, I would say that you get an output which mainly contains gene clusters encoding for terpene substances, type 3 polyketide gene clusters, alkaloids, and so on, because these are more substances or gene clusters that are more abundant in plants. In bacteria, you may get just another output also on gene clusters encoding for non-ribosomal peptides, type 1, type 2 polyketides, for example. So I hope I could answer that question.
Okay. Yeah, thank you, Yvonne. If there are any questions again for the both speaker, please raise your hand or write it in chat box. Maybe I will ask Professor Claudia Terribili. Does consuming too much fiber also causes liver damage?
Yes. I mean, the point is not what you consume. The point is what the amount you consume and the calories. I mean, if you eat, let's say, a kilo of bread, the amount of calories is much higher than a kilo of salad. But of course, the taste of 1 kilo of bread is much better than the taste of 1 kilo of salad. And if you put some butter on your bread, the calories increase. So I think we need to make a balance of our daily behavior. I mean, if we eat more calories, we need to spend more calories. As I told you during my presentation, our weight and our metabolic functions is like the bank account. I mean, if you gain a little amount of money, but you are so able not to spend, you're getting rich. But if you gain a lot of money, but you spend you spend more, you get poor. So everything is balanced. We need to be careful. And then the basic point is to teach our children to behave properly. And our children, at least in Europe, don't behave properly. They are in front of a screen. They don't move. They don't go to walk. They don't go to play football. They don't go on the bike. They stay home, and this is bad. That's why we're having a real problem with adolescents.
Okay. My next question is there any effect of a supplement on the liver health?
You mean the supplements?
Yeah, like vitamin?
Well, I mean, the supplements have never been demonstrated to be helpful. The supplements are very helpful for the companies that are making the supplements. They're making a lot of money they're making a lot of good for the producers of the supplements, but there is no evidence that they're making good for the liver of the subjects taking the supplement.
Okay, thank you.
So take a message, don't take supplements.
Oh, okay, thank you for your suggestion.
Eat the fruit, it's more than enough.
Oh, I see.
You're lucky enough to live in a country like Indonesia, where the fruit is so beautiful. And we have mangoes, you have everything.
Yeah.
Why don't you have to take supplements? If you're living in Europe, I mean, you go and then eat an apple or an orange or a pear, and that's it. It's better, it's cheaper, it's much more healthy. Trust me.
Yes, of course. Thank you for your suggestion. Okay, if our time is left 2 minutes. Okay, or Yvonne, do you want to ask the question to Professor Tripoli?
Yeah, actually, I would also want to ask a short question to Mr. Tiribelli. I wondered if there are any numbers. What is in general more harmful if one drinks too much alcohol, or using too much calories from early days on?
I didn't touch alcohol for obvious reasons, because alcohol, there is a safe amount of alcohol daily, which around 30 grams per day, both in females and males. 30 grams of alcohol per day means half a liter of wine or a small glass of super alcoholic schnapps or whatever it is, which means that if you have one glass of wine with your meal, it's good. What is bad is to drink outside the meal, because the absorption of the alcohol without any aliment is very quick. This is why the heavy drinkers are used to eat together with alcohol something which is oily, like olives and peanuts, because you have a sort of film of oil in your stomach, so the absorption is less, and then you can drink more. That's the trick they're doing. So, I mean, a limited amount of alcohol is safe, actually. But, I mean, it depends on how limited is limited. And, I mean, a good glass of wine is good. And let me tell you that, unfortunately, I'm a teetotal. I cannot drink alcohol, not because I don't want, because I cannot stand it. And this is my limitation, because, I mean, with a good meal, a glass of wine, assuming that your religion is allowing to do it, it's good.
Okay, thank you for the answer. There's 3 questions again in the chat room. First, for Professor Tripoli. The question is, as we are getting older, is it true that our metabolic rate are also slower than when we are young?
Yes. Yes, the answer is yes.
Yes, okay.
That's it.
Yeah. And there's 2 questions for Yvonne. Did you also conduct any research in Germany on cool culture to activate gene cluster for the production of natural products? Based on your experience, which substances from terrestrial and marine ecosystem produce more new metabolite at this time?
Okay, concerning question one, we didn't do co-cultivation approaches, and I tell you why. Because later on, the identification of the produced metabolites is just too hard. If you do co-cultivation, you may activate something, but you do not know what is produced, and then you need to go again into the lab, isolate the substance, do chemical analysis to really identify the produced substance. We had the attempt to do a targeted activation, because if we use these kind of regulators, then we exactly know where they bind, which kind of antibiotic gene cluster is activated, and this facilitates later on the identification of the compound. And therefore, we didn't do co-cultivation, which also may work, but we only focus on this targeted activation approach. So the second question is, which kind of organisms do produce more antibiotics, terrestrial actinomycetes or the marine-derived ones? We cannot say until now. You can have marine organisms with really a lot of antibiotic gene clusters, but also terrestrial ones can harbor more than 50. So I don't see a difference in terms of the potency of terrestrial against marine organisms. So there's still enough out there to be identified in terms of new secondary metabolites, I would say. Okay, but I think there's an additional question. Eva, would you like to?
Okay, I think there is no any question again. Okay, I think time is finished. Time is the end for the question. Finally, we come to the end of the seminar. I want to draw a conclusion from what speaker said. From Professor Ivan Mas, we can learn that a self-guide strategy can be used to activate antibiotic biosynthetic gene cluster in a targeted manner and thereby exploit the biosynthetic potential of actinomycetes. And from Professor Tribeli lecture, we can conclude that excessive sugar and fat consumption from a young age can lead to the obesity, leading to the development of metabolic associated fatty liver diseases and non-alcoholic fatty liver diseases in the children. Unfortunately, the development of metabolic associated fatty liver diseases in the children is faster than in adults, and efficient therapy to treat this disease doesn't yet exist. Hence, the best options still depend on the lifestyle changes. Okay, finally, I would like to thank to Professor Ivan Mas and also Professor Claudia Tribeli for their informative and interesting presentation, and all the participants for their very active participation. And now I will hand over the event to the MC. Thank you. Ms. Aulia, time is yours now. Thank you.
Terima kasih.
Terima kasih banyak.
Thank you, Professor Tribeli, Professor Ivan Mas, and also Dr. Ira for the amazing and also fresh sessions this afternoon. Distinguished guests, ladies and gentlemen, now we have arrived to the end of today's conference. On behalf of official committee, I would like to thank you for your attendance and also active participations on the first day of ISIBio 2022. We're signing off. See you on the second day of 9th ISIBio tomorrow at 9 AM Jakarta time with another interesting speakers and also materials. Stay safe and also healthy.
Bye-bye. Thank you.
Thank you very much, Yvonne and Professor Tribeli.
Thank you. Bye.
Bye.
Bye-bye.
Bye-bye.